Hello, I'm your host, Ray Dogum, and welcome to Vibecast. Thank you for joining us as we explore the exciting advancements in technology-enabled collaboration to excel important drug development. VibeBio seeks to find every cure for every community. We think big, as no one should be left behind in the pursuit of living a healthy, happy, and productive life free from disease. Collectively, we have the skills, we have the technology, and we have the passion. We now need the community catalyst to bring it all together. That's Vibe. We see a future where biopharma experts and patients collaborate to identify high potential medicines, have the ability to access capital on demand to develop them. The Vibecast is our weekly informational podcast where we explore some of the hottest topics in drug development and technology innovation with some of the dynamic people that make up the Vibe community. Join us to learn, imagine, question, and help us identify solutions together. Joining us today is Dr. Howard Berman, founder and CEO of Coya Therapeutics, as well as Daniel Barvin, Vice President of Operations and Patient Advocacy at Coya. Dr. Berman will be presenting on neurodegenerative disease and the research that Coya has been doing. Welcome, both of you. Really appreciate you joining me on the show today.
Thank you. It's a pleasure being here.
If you wouldn't mind quickly also sort of giving the audience a more detailed background on both of yourselves. So maybe we could start with Dr. Berman, and then we can go to Daniel.
Wonderful. Pleasure to be on the call today. Thank you very much for your time. My name's Howard Berman. I'm the CEO of Coya Therapeutics. Brief background on myself. I'm a scientist by training, doing a PhD at Cornell in New York City at the medical school there. I then went into biopharma, and I spent many years at different companies commercializing, developing, and launching drugs in a variety of different indications, immuno-oncology and neurology. And I'm also an entrepreneur. I'm on the board of a digital health company. And having started Coya Therapeutics a few years ago, I am very excited about what we're working on. And I've had the pleasure of being able to hire great people, including Daniel Barvin, who was actually employee number one. So with that, I'll let Daniel introduce himself.
Thank you so much, Howard, for that. And Ray, a pleasure to be here. And thank you all for listening. My name is Daniel Barvin, VP of Operations and Patient Advocacy. So I joined Coya as I started my MBA. And at the beginning of that educational process, I lost my father to frontotemporal dementia. That experience was incredibly traumatic. And the last line of generational trauma, as I unfortunately lost my aunt and uncle to ALS previously, that loss really propelled me to find a mission to advocate and work in the space of biotech. And post MBA, I started an organization focused on fighting for awareness and education and resources for the familial ALS and FTD community. And after spending about 2 years creating that organization, Howard found me and wanted me to join Coya, as he needed someone who would be incredibly passionate and have the energy and focus that it takes to be inside of a biotech startup. As you'll see, that it has not been an easy journey, but we are hoping to really change the world, and nothing that changes the world is easy. So I'll leave it there, and I think that we have a great story to share with you. And pass it back to you, Ray.
So let me give you a little overview, and this is a very high-level overview, and I'm going to try and make this very simplistic for the audience to understand on what happens in neurodegenerative diseases and how inflammation is playing a critical role in the disease process. On the left, this is a schematic on the various neurodegenerative diseases. As you see, Parkinson's is driven by dopaminergic neuron dysfunction. ALS is driven by motor neuron degeneration and dysfunction. And Alzheimer's disease is driven by pyramidal neuron dysfunction. And they all happen in different parts of the brain. Whatever the cause, the underlying cause, and there's numerous causes of neurodegenerative diseases, how they are initiated. And I can tell you that no one really understands the multiple, it's a multifactorial process. The next step that happens in the process universally is that the neurons, when they become sick, the immune system sends immune cells to the sites of the damage at the neurons because they realize that there are issues at the neuron. And initially, it's successful in terms of removing toxic proteins, misfolded proteins and other aspects of the damage. But eventually, the neurons are not getting fixed, so they tell the immune system, in essence, take me out, kill me. And the immune system is ramped up and results in those neurons dying. So it leads to things like apoptosis, necroptosis, ferroptosis. There are various ways of cell death. So you end up with dead neurons. So the immune system is a critical feature, a critical mediator of this process, this neurodegenerative disease process. And one of the most critical cell types in the body that plays an important role, and which we have discovered in our laboratories, are the cell type called the Tregs, or the regulatory T cells. The Tregs are the most important cell in the body that controls inflammation. And in many of these neurodegenerative diseases, the Tregs are dysfunctional, they're compromised, they're not doing their jobs. So you get a seesaw at the bottom where you have more pro-inflammatory cells and less functional Tregs. So you have a haywire system with ramped up immune response, and unfortunately, the patients decline relatively rapidly. So what are Tregs? On the left, you see in blue, these are an anti-inflammatory cell types, and they inhibit the pro-inflammatory cells. And that causes, at the bottom, we call it homeostasis, which means that the system is in balance between anti-inflammatory T cells, Tregs, and pro-inflammatory cells. Unfortunately, on the right, in neurodegenerative diseases, also in autoimmune diseases, you've got messed up Tregs, compromised Tregs. And unfortunately, that results in heightened inflammation and a runaway immune response and degenerative disorders in these conditions. This just illustrates, and this is a very technical slide, but I want to just show you what's happening. In neurodegenerative diseases, you have dysfunctional Tregs, and we have a drug called Coya 301. It's a type of protein that when you inject it into the blood, into the subcutaneously, it enhances these Tregs, it ramps these Tregs up, and that's what we want to do. We want to cause those dysfunctional Tregs to become functional, to enhance and to increase the numbers and the function. And in the blue circles here, we illustrate that in the bloodstream, what they act to do is they block the types of pro-inflammatory cells. They're called the T effector cells. So they robustly inhibit these pro-inflammatory immune cells. And less robustly, they inhibit a type of cell called the myeloid cells. These myeloid cells also play a role in this inflammatory process. And what we've learned is, in order to be likely the most efficacious, one has to ramp up the Tregs with Coya 301, but also find a way to inhibit these innate myeloid cells, these pro-inflammatory cells that are only inhibited by about 20 to 30% of these Tregs. So we have discovered another drug that when you give it with Coya 301, acts to inhibit these myeloid cells, so they have synergistic effects when you combine 301 with this other drug. And that's called Coya 302. What Coya 302 is it's a low-dose interleukin-2, which acts to decrease the dysfunctional Tregs that I mentioned. It increases these Treg numbers and suppressive function. And CTLA-4 IG, this is a fusion protein that acts to reduce those myeloid cells that I discussed with you. It increases the anti-inflammatory macrophages. These are a good guy type of cell, and reduces all of those bad guy pro-inflammatory cytokines. So going back to the previous slide, instead of the 20 to 30% suppression, you get much higher suppression of these myeloid cells. You still are getting the robust suppression of these T effector cells. So now you have a synergistic mechanism effect between 301, which was low dose IL-2, and the CTLA-4. And that's called Coya 302. So we have run Coya 302 in a study in ALS patients, which is, as you know, a very terrible disease, which is characterized by motor neuron dysfunction And these patients unfortunately decline month after month after month. And at 6 months, typically, the patients are declined so much so that sometimes they can be at the start of the trial in walking, and at the 6-month time period, they can be in a wheelchair. So anything that can slow or even better the disease would be a remarkable feat. We conducted 302 in a trial, which we presented at the MDA conference. Now we looked at 24 weeks as well as 48 weeks. We went even further than what you typically do. And guess what? We stopped disease progression. The patients did not decline, which was a very exciting outcome. We also showed significant Treg function enhancement and Treg numbers, which is what we wanted to see. And we we decreased the blood biomarkers of inflammation. And so far, the therapy was safe and well tolerated. Now, we also are presenting data for just the Coya 301, which is a low-dose IL-2 in Alzheimer's disease. And we're validating that, in fact, Tregs, when you enhance Tregs by itself, whether they can have an impact on Alzheimer's disease patients. We're going to be presenting that data next week at the Keystone Conference. And this is going to be on May the 16th. And we're excited to present that data.
Before you continue, maybe I can ask you a question about ALS and Alzheimer's and how they, what's their connection? And you kind of mentioned it, I think, in one of your first slides about how these different diseases manifest themselves in the brain. Can you dive a little bit deeper and maybe talk about, the patient's experience as well?
Sure. In whether it's Alzheimer's disease, whether it's Parkinson's, whether it's ALS, inflammation is present. And inflammation is present not just in the brain, not just in the central nervous system, but also in the peripheral blood system. So the most critical thing to be able to accomplish here is to reduce those levels of inflammation and what's called oxidative stress. So the diseases are very different. ALS is very different than Alzheimer's. The presentation of the patient is very different. ALS, the patients decline much more rapidly, and they are really characterized by motor dysfunction, and they're cognitively usually intact. Whereas in Alzheimer's disease, it's a disease really of the central nervous system of the brain, and unfortunately, they are cognitively impaired, and the decline is usually slower than in ALS. But universally, inflammation drives the disease process, we believe. And that by being able to inhibit that inflammatory cascade or the inflammatory process, we think that we can make a significant difference in the patient's or the decline of the patients. Okay, this is the decline in this slide of the patients in ALS. Typically, based on the PROACT ALS database, which is a large database that has been quantified over many, many years, patients have been found to decline on a monthly basis by differing rates. As you see here, every month, the patients are typically declining, the average patient, of about 1 point per month. So at 24 weeks, on average, plus or minus a standard deviation, patients are at about -6 or -7 on a scale called the ALS-FRS score, which is a universally accepted scale which documents the clinical function of the patients. The most recent drugs that have been approved, the most recent one actually, called Relyvrio, showed a decline on the ALS-FRS score at 6 months of -6.64. The placebo was almost -10, and Relyvrio was approved in ALS. Radicava, which is another drug that was approved prior to Relyvrio, showed a decline of -5 points on the ALS-FRS and a placebo of -7.5. So it is clear that therapies approved are only slowing progression in a very slight manner. They do not stop progression, unfortunately, and the patients continue to decline incrementally on a month-by-month basis.
What is a typical ALS-FRS score for, someone with severe disease presentation? Is it like 10, negative, 10 or 20? What's like the value?
Yes. Well, there's no one answer, but I can tell you the scale, which goes from 0 to 48. Typically, what you see is that patients below, and we use this in clinical trial stratification, patients that are below 30 and certainly getting into the 20s are much more progressed than patients that are above 30. So it is very clear to us that the sooner that you start treatment, the better, at least for patients who when you stratify for clinical trials, because at that point, the level of inflammation and potentially oxidative stress is much higher.
Interesting. Thank you.
This is what we, in our clinical trial, which we ran for Coya 302, this is what we showed. These patients were declining month upon month. These patients were, on average, declining at about 1.1 points per month prior to treatment. So prior to the start of the treatment, the baseline, let me just say, at start of treatment, the baseline on average for these 4 patients was 33.5 points on the ALS-FRS. So they weren't declined below 30, as I just mentioned to you, but they were not, certainly they weren't above 40. So they did have certainly on average their disease was more advanced than a newly diagnosed patient. But what the most exciting thing here is that the patients stopped progression out to 6 months. So they were actually slightly better on their ALS-FRS score at 33.75. And at 48 weeks, they had only slightly declined to 32. And these were not statistically significant in terms of decline, either at 24 weeks or 48 weeks versus the baseline, showing that this treatment actually stopped progression. I will say that this was an open label study. It was in a small cohort. It was very promising, but we did not do a placebo versus an active arm. So we're not making any claims, but certainly it's very exciting for us. And it's going to lead to doing additional studies, a much larger double-blind placebo-controlled trial with this therapy.
That's excellent. Do you have a plan or timeline for this next study?
Yes, and I'm going to get to that in 1 or 2 slides.
Excellent.
Thanks. All right. So this is the next study. This is what we're planning. This is a phase 2 study design, and now it's subject to change. We're working on biomarkers, which we've established, and we've identified specific markers of oxidative stress and inflammation that are mediated by this therapy. So can you imagine being able to give the therapy and being able to track the successful or the efficacy of the therapy by looking at levels of certain biomarkers in the blood? That would be what we think to be a remarkable feat. So those analyses are currently ongoing. We're planning on doing a primary efficacy endpoint, which looks at a number of features, including the ALS-FRS. We're going to be looking at patients that are on average over 35, which is to one of your questions about the degree of decline of the level of dysfunction of the patient at baseline. We want patients that are not as far declined in this study. We're planning on filing this IND with FDA this year, but towards the end of the year. And this study should be about, we anticipate, between 14 to 16 months to enroll and to complete the trial.
That's exciting. Do you have any feedback from the FDA on the study? Have you, I'm sure you've had meetings with them, but is there any sort of feedback they provided you that you're willing to share here?
We are going to be receiving our pre-IND feedback probably in September of this year. We also are looking at, so Coya 301, Coya 302 is Coya 301 plus another drug called CTLA-4. So we think that Coya 301 serves as a backbone treatment, meaning you can combine Coya 301 with multiple other drugs that target other pro-inflammatory cell types. And that gives us the opportunity to partner strategically with big companies and smaller companies together. Thus, we have opportunities from a business development perspective. And this slide just illustrates that. Coya 301 combined with CTLA-4, which I just mentioned, is Coya 302, and we've shown therapeutic benefit in ALS patients. But imagine we can combine 301 with drugs that block cytokines, things like IL-6 inhibitors, TNF-alpha, IL-1, beta inhibitors. We can combine with other drugs called complement inhibitors, or ferroptosis inhibitors. So the sky is the limit in terms of the ways that we can use 301 to broaden our collaborative nature with companies. And the reason this is important is that the immune system is redundant. There are many different pathways involved. And if you block one, then another one can take over, which we've seen in cancer and oncology, that you have to block multiple pathways in order to be very successful. And also, much like HIV and AIDS, we believe combination therapeutics are the way of the future. So that's why 301 really serves as a base adjuvant backbone treatment with other inhibitors. And we're excited in terms of our partnership strategic opportunities that exist with pharma companies.
That is interesting. So I know in the past couple of years that the FDA has approved, I believe, 2 drugs for ALS. And can you discuss like what has brought them to that point where the FDA, has identified the data that proves, or at least has enough evidence to show that it's worth having the public start to use them? Can you kind of describe that? And are those drugs also building upon themselves the same way Coya is, or can you just describe a little bit more?
Right. This slide, which I showed before, illustrates that. Relyvrio, which is a combination of 2 generic drugs, which was approved by the FDA last year, which was based on a phase 2 study of patients with ALS. They showed a decline of -6.64 points, as I mentioned to you before. But I can tell you that they've taken the approach that they're not targeting inflammation per se, they're targeting the mitochondria, the endoplasmic reticulum, and they are trying to attack the disease in different ways. We think, though, however, that our drugs, our 302 in particular, will be complementary to Relyvrio and Radicava because they all target different ways of addressing the disease. We're targeting the inflammatory process. So there may be opportunities, and likely, in terms of combination strategies with Relyvrio and Radicava. But I can tell you also that the FDA has really been proactive in understanding the patient advocacy community because there's nothing for these patients, really. There's very little to help these patients, and they're declining month after month. So Relyvrio, or Amylyx behind Relyvrio, has been very active in terms of bringing their data to the FDA and getting the advocacy and support of the agency in terms of their data, and also what they're planning to do in terms of once it has been approved, their confirmatory study. So the data has built upon itself, but it's also liaised with the patient advocacy community to really move the needle with FDA. And I think we're moving into a very supportive regulatory landscape with FDA at this point. So we're at the right time and the right place.
How is it working with the FDA in terms of trying to keep patients in mind at the same time? Like you said, they're more inclined to be receptive to new treatments now, you're saying? And I'm just wondering, because there's these 2 drugs in the market, is that creating more or less opportunities for Coya and other companies like yours?
I think it's creating more opportunities. I think the more drugs that are out there that can combine different mechanisms, we think that combination therapies, as I mentioned before, are the way of the future. That this is a multifactorial disease that relies upon many different pathways, and that any successful treatment, any successful long-term durable treatment will require the whole host of armaments that are out there. So we think that this is a trial. When we run this trial, it will include the approved drugs as well in both arms.
Also, how do you know which patient should be getting the different combinations, right? Is there some sort of diagnostics journey that a patient would have to go through? And maybe Daniel, you might have more information on this, but yeah, anyone, Howard or Daniel?
No, I can take that.
Awesome.
Both arms, whether it is there's 2 types of ALS. There's a sporadic, which is the majority of ALS patients. These are not driven by a defined genetic mutation. And then there's the lesser, the 10% that have mutations like the C9orf72, the SOD mutation. So we're going to include all patients, 100% of the patients. Now, both arms of those patients placebo and active are going to have the same, they're not going to be different. So they're both going to include the standard of care drugs, whether it is Relyvrio or whether it is Radicava. And so there's not going to be any differential between both arms. There is going to be a different work. Everyone's going to have the genetic testing, and we're all going to understand the type of ALS, whether it's bulbar or whether it's the limb onset. So those are all standard ways to quantify ALS, but the arms are not going to be different in terms of therapeutic modality.
That is interesting. So, I was thinking about why it's taken so long to get these first drugs approved. And I was wondering whether it was just the discovery of the science and the chemistry and the biology, or was it more a regulatory issue in terms of like how effective they are? So is it our understanding of ALS and Alzheimer's and these neurodegenerative diseases, or is it the availability of good treatments?
Yeah, no, unfortunately, the availability of all the treatments have not worked. And the FDA requires a double-blind, randomized, placebo-controlled trial. And unfortunately, none of these drugs have been efficacious. And you cannot separate the placebo from the active arms statistically. Unfortunately, that's the reality of the situation. And it's the same in other trials, in other indications like glioblastoma and pancreatic cancer. There's just no good treatments. What I can tell you is that now that we understand the disease process better, and we really feel that inflammation is playing a critical role, and oxidative stress is playing a critical role, that this is a new era, much like in cancer, much like immuno-oncology transformed cancer. We think that we believe that immunotherapies on the other side in ALS are going to change the way that ALS is treated. But we also think that combinations are the way to go. Many companies are targeted on one pathway. We don't think that's going to be as good as targeting multiple immune pathways.
Agreed. And like you mentioned, it's the same in oncology as well. There are many pathways, and combination therapies seem to be the current and future state of affairs. So that's really interesting. And that makes sense, more broadly speaking, if you think about it. The body is so complex. Many things are happening all the time. And we also sort of rewrite how that's happening in some ways with our environment. So it's quite interesting how that all plays together. Yeah, that's really cool. So can you describe the upcoming clinical trial in more detail, and what kind of patients may you be looking for? What's the inclusion/exclusion criteria? Maybe patients listening to this might want to be involved. So what would you tell them?
Well, first, I want to say we're going to have a trial that includes multiple sites, likely around the country, because we want to make sure that we enroll in a time frame that is reasonable. We want to have of course, the leading thought leaders and those distributed around the country so that patients have access to this trial. We have not, while we're working on the various inclusion and exclusion criteria, those are the criteria that requires how a patient can be included or be excluded from the trial. We think that likely this will be all patients. So whether you have a genetic mutation that's driving the disease or whether you don't have a genetic mutation, this trial will be available. The therapies likely will be subcutaneous injections so that the patients can have this administered in the home setting. So the caregiver can simply inject it like an insulin injection. And so that makes it very easy for the patients. They don't have to get complex infusions, wait around in the office. This makes it a much more simplistic manner to have therapy. And so we're excited about that. And we, in terms of the patient status, we think that this may be stratified by patients with specific biomarkers. And we're working on that currently. Whether they have high levels of inflammation and oxidative stress may be a factor in terms of entry into the trial, and also their baseline ALS-FRS scores. And those should be fleshed out in the coming weeks.
Excellent. So you mentioned oxidative stress a few times already. Is there, in addition to therapies, drugs, treatments, is there anything else patients can do to reduce oxidative stress in their entire being? What has the evidence shown around that?
Well, the evidence is growing, significantly, and it's being led by Dr. Stanley Appel at Houston Methodist. Namely, that what oxidative stress, essentially it's free radicals, and everyone's heard about free radicals. Things like 4-HNE, oxidized LDL. These are markers of what we call oxidative stress. They are very toxic to the neurons. They can cause the neurons to undergo death, and they go hand in hand with inflammation, typically. There is a very tight correlation that we're beginning to find between the level of oxidative stress that you can measure in the blood and the degree of decline of the patient. So the higher the level of oxidative stress, the faster the patients decline. And in fact, what we're beginning to see is you can actually pinpoint their ALS-FRS score with the amount of oxidative stress that they have. That data we're evaluating that data. I think some of it is genetically driven. It's part of the immune response, and it's also part of the disease pathology or the pathophysiology of the disease. It's a complex issue. We don't really know how you can modulate oxidative stress naturally or what you can do, but probably lowering inflammation and doing whatever you can to reduce the level of inflammation will benefit the level of oxidative stress. But certainly we're excited about this. It's a new area, and it's an evolving area that's many publications are being brought on this subject.
Interesting. What does the funding landscape look like for biotech companies these days, and particularly for ALS and other neurodegenerative treatment companies?
Well, previously it was
you mean previous to COVID or during COVID?
Even a few years ago, there was a lot of concern about funding neuroscience companies because of all the failures. Alzheimer's disease has been a landscape filled with failures. ALS has been filled with failures. These neurodegenerative diseases are very complex, and with the emergence of the immune system and the role that it plays in these diseases, I think it's become much more exciting. And specifically with all the new data in Alzheimer's disease, with Eli Lilly's drug and Biogen's drug, that's evoked a strong sense of interest by funders to move into the space. And furthermore, what's happening in ALS with the recent approvals, with the positive regulatory landscape, I think also has shifted the sentiment. And now we're in a very conducive environment for funding. So times have shifted, times have changed. We are where oncology was, where everyone was starting to put money into oncology. People are really starting to pile money into the neurosciences, particularly in ALS and in Alzheimer's and these diseases. So I think we're in a very unique position, and it's very positive for our company and others in the field.
That's excellent. Can you describe some of the relations you have with patient advocacy groups, and just describing how you work with them, what they do for you, what you do for them, sort of deal? And just be curious to learn more about that.
Yeah, Sure. I'll let Daniel talk to that.
Sure. I think that we are a bit preliminary in terms of connecting with patient advocacy groups at this moment, as we don't want to overpromise and underdeliver. So our goal is to continue forward with this trial. And as we have understanding of the inclusion-exclusion criteria, start bringing awareness of our company and our trial to patient advocacy groups like INALS, the ALS Association, et cetera, to help spread that message and help us recruit and inform patients to ensure that they're aware and have access to the trials that we're about to put forth. I do think that the patient community can be very powerful in terms of bringing up just a fervor for the therapies that we are about to deliver. You look at the patients who are suffering from ALS, and in the past and currently, those people have died in 2 or 3 years. And so it's truly a yearning for something that will change that dramatic course of suffering. And so when we have something like the proof of concept data that we released, I think that patients will want to enter our trials, and we hope to be able to provide as much information and access to those as possible. I think that the hardest thing will be there are only so many spots, and we wish that we could give this to as many patients as possible.
Excellent. That sounds great. Thanks for sharing that. I'm also thinking about what are some of the risks you might have identified during your initial proof of concept trial or test. Anything that you can share there, and
Yeah, I think we've reduced a lot of the risks in terms of drug development, and I'll tell you why. A lot of drugs are new chemical entities. They require, one doesn't know the toxicity, one doesn't know the immunogenicity, which is how the body is reacting to the drug. There's a lot of safety issues even before you look at efficacy. So I think from a safety perspective, we reduced that risk dramatically. The low dose IL-2 which is proprietary and unique, looks to be very safe and well tolerated, and we know IL-2 because it's been around for many years, at least in a high dose format. The CTLA-4 Ig is a drug we licensed from Dr. Reddy's, which is a $1 billion company. It's a proposed biosimilar drug. And CTLA-4 has been known as abatacept or Orencia, which has been marketed by Bristol Myers Squibb for rheumatoid arthritis. So we know what the tolerability profile is there. So that risk has also been reduced. So from a safety and tolerability standpoint, we know what we're dealing with. I think from an efficacy standpoint, we didn't anticipate the results that we saw. We didn't anticipate that we could stop progression, at least in these patients, and that the blood biomarkers would correlate, the Tregs would jump up. Now I think it's an issue of execution, is taking these, moving these through with the FDA, getting the FDA's approval on the trial design, on our manufacturing. I think that's the next step. And we're actively working. We're confident. We know what we need to do. We've got experts. We got professionals. And so we are so far on time, ahead of schedule, and we're going to continue to meet those standards.
Excellent. Howard, Daniel, I just want to thank you so much for joining me today on Vibecast and talking about neurodegenerative diseases, ALS, Alzheimer's, and also Tregs, and explaining that concept to the audience and community as well. Really looking forward to your progress with this new drug, Coya 301, 302, 303. Hopefully those will all sort of continue down your pipeline. So that's exciting. And where can people reach you if they're interested in learning more?
Sure. They can go to our website, which is Coya Therapeutics, Coya Therapeutics.com. There is also lots of information on our website about our science, about what we do, how we're approaching this problem, all about regulatory T cells and Tregs. And we are also, we have a stock symbol because we're a NASDAQ company, and we're COYA as a symbol. And I'll ask maybe Daniel if I missed anything about how to find more information on this.
Sure. So I think that often we get patients who request information about how they can learn of upcoming trials. And I'll say that the best way to do so is through our social media networks via LinkedIn, Twitter, things of that sort. As we continue to progress in terms of pursuing the next clinical trial, we will have notices put out, updates, PRs, and that should be informative to the patient community.
Yeah, I will say this last bit of information, this is a very personal mission for us, and I'm talking to the patients. We know the difficulties of having a neurodegenerative disease. We've all been afflicted. I've personally had a family member who passed away recently from a neurodegenerative condition. Daniel, unfortunately, has had the same experience, and he has a genetic mutation. So we need to move quickly. There's no time to waste. This is a serious disease, a series of disease conditions, and we're all personally vested. We want to make sure that this moves forward quickly, timely, and expeditiously.
I totally agree. And as our population continues to age, this is going to become more and more of an important issue. So I'm totally glad that you're working on this, and glad there's communities getting built out around ALS, Alzheimer's, et cetera. So fantastic. I'm looking forward to sharing this with our community.